So thank you everyone for listening and inviting us. And as already introduced, so I'm leading the Horizon project deCYPher, which focuses on deciphering cytochrome P450 enzymes by digital tools to produce these flavonoids and terpenoids. And yeah, we want to move away from these traditional production methods to more bio-based production, where we want to increase the development or fasten the development time by really introducing AI and machine learning in the design and learn steps of the traditional design, build, test, learn cycle in synthetic biology. Why? Because, well, traditionally, it's quite a slow production process. You are depending, of course, on the cultivation, and it's also not always that healthy. And by using microbes, and I guess I'm here in the community where we really believe this, that these microbial cell factories and bioprocesses can give a good alternative. And so we don't only want to rely on the data that we get from researching the plant and identify the plant pathway in the lab, but also by the data that is already out in these databases on plant pathways, and having a smart way to look at this data. Just to give some examples of products that we are focusing on. So one class are terpenoids, in which nucatone and pyrrolic acid are two products that are currently not on the market or partly on the market, and have applications in food and as a flavor and a fragrance, but also in PET plastic, for example. And then the other class is on flavonoids, and these also have food and pharmaceutical applications. But in all these plant metabolites, these CYP proteins are key to get to these more complex molecules, which these specific bioactivities. So what do we want to do? So we want to enhance our DBTL, and we identified 5 challenges. So one is that we need a more efficient identification of these CYP enzymes, where we think a digital tool could help. Most of the time, the digital tools today are not fit for the biotech application as such, because you have not that much data or it's noisy. And then that was already hinted to, you need infrastructure for the metadata management and processing, especially if you want to do AI and machine learning. We believe that an integrated holistic approach that we can map from identifying the enzymes towards the cell factory engineering and even the upscaling to bioreactor levels should be all being integrated. And then, of course, we also work on identifying the needs of the stakeholders so that we can move faster to the market with our products. And so these enhanced DBTL cycle, we will want to introduce through creating these different digital tools. And you can see these go from different use cases, from enzyme level to cell level to process level. But for all of them, to make it easy, we want to create a kind of a plug and play generic pipeline so that you can actually apply it to any kind of biotechnology problem that you encounter. To do this, we brought together a strong integration of AI and ML and Symbio, together with then Elixir for data management infrastructure, but then also groups that are strong in open science and citizen science engagements, like Biofaction, also partner in Bioindustry 4.0 and the BioArt Society. So for that last part, we do also quite a lot of outreach, like stakeholder engagements, where we will organize roundtables and citizen panels. But we also work with artists, and that's also where probably a collaboration with Bioindustry 4.0 can exist. But as said, what would be interesting, or what you asked me to do, was to tell a little bit more about our data management and the reinforcement of the tools. And so I highlighted 2 work packages where I think that data is important, or how the data is, and where maybe further collaboration or new collaborations can exist. One is on work package 1, where we are creating this generic AI pipeline, and then the work package on data management and integration. So the overall goal is here that actually in this design, build, test, learn cycle, that we really implement AI in the learn and the design component, and that it is an active learning, so that it can help us even with when you have not that much data, that you can actually start predicting which data you should generate to learn more and to go faster through this DBTL cycle. And for that, we identified 3 use cases. One is on protein work. And so this could be, for example, a question that we have: how can we easily find proteins with a specific functionality based on, for example, public data sets that are available, or then a limited set of tests that we do in the lab to characterize a type of protein, and then using this data generate or propose new enzymes to be tested to further enhance our digital tool, but also then faster identify the protein with the desired functionality. A second use case is then the strain optimization. And so here it is about the molecules with the novel production host, because we are not working in model organisms. And so here the AI should help us then to identify how the architecture, for example, of the pathway, which building blocks, which enzymes, which promoters to be used within the pathway should be combined to get better production. And then in the last use case, it's more focused on process optimization, where we will use the data from small-scale lab experiments and transfer it, or make sure that it can be translated to larger scale production, and see if we can fast that also. How do we do this? Well, through a multi-step process. And so you can see you have now the 2 worlds, the wet world, or the world of the molecules, where we do the building and the testing. And so that's the setup of the experiment and the performance of the experiments in the lab. But then we have the world of the bits, where we have the learn part. And in that one, we have steps where we do data indigestion. So we bring in data, we can do feature calculations, and then you can choose depending on the problem that you want to solve. For example, it could be like a modeling of the protein, or a feature that you extract, like the amino acid composition, and so on. And with these data, you will do a model training, which then also can feed into a model prediction. For that, we can choose between different models that are being linked within the toolbox or the pipeline. Using that, we can go to the design where virtual experiments are being generated from these features are again calculated to do the model prediction. And based on that, the digital tool will then propose a selection of experiments. So at this moment, we already built the generic toolbox. We have the different models, and we already, for the first time, run it on one specific case, which was then an expression or functional expression of these CYP proteins. Because these CYP proteins, they need also transmembrane domains, and so we created new domains through this digital tool, which will now be tested. But you can see the interface is quite light, and that's also something that we want to make sure that actually also bioinformatic experts can actually use the tool. So the wet lab people can easily also use the tool by having it in easy steps, bringing in the different elements. Of course, to do this, we need a good data management and integration. And that's why we really have a specific work package only dedicated to this, where the overall objective is to make sure that we govern the data flow or the research outputs from the wet lab to the dry lab and vice versa. So actually, the role of this work package is just to enhance this seamless data integration between where the data is being generated and collected. And that's in 3 work packages that we have, towards where then the data is being processed and used in different algorithms in this data pipeline. As already said, this can be very challenging because you need to integrate wet lab data sets, and we are with 10 different partners. So each of them have their common methods of annotating the metadata or using different software. Also, metadata transformation can be challenging because the different various metadata sources that are needed for the AI may require ad hoc transformations and additional information that we didn't have or that is now necessary. And also to make sure that we have an impact as much as possible when we communicate, we want to make sure that this metadata is also ready for publication and that they also meet these standards for the different repositories that you have. And so you can see that's also why in our per chart, you can see that the AI is in the middle or surrounded with the wet lab work packages, but this is all supported by our data management. For that, we try to implement a common metadata framework and data flow. And so we identified 5 tasks for that. One is, of course, the data management plan, but more importantly, all partners from the wet lab and then the dry labs, they dedicated a person, at least one person who is in a data management working group, who is also now, because it's the start, during the start of the project, it was more intensively, is coming almost 2 weekly together to define how this metadata framework had to look like. For that, they also looked or assessed what were the current internal practices of all the different groups and what is needed and what is required of metadata from a a practical point of view, and then also from all the different aspects where we want to implement it. Always taking into account that we wanted to make the workload as less as possible, and that it should be easy to do everything, because otherwise, if you have a very big and log structure, then it will be cumbersome to add data to the system. And then these give then the input for actually defining the metadata standards, how we will, the models, how we exchange data, and actually creating this data framework. And of course, then once we have data, we can deposit it. And it's actually in those two last parts where we really rely on resources that are provided by Elixir and that are also being extended and improved for our needs within the deCYPher project. And so for this, we have two partners, Elixir Spain and Belgium, which are hosted by the VB Data Core and the Barcelona Supercomputing Center. And so you can see already here some resources from Elixir that are already existing, but that still had to be improved for us. So more specifically, in deCYPher, there are two main resources that we are using. One is DataHub, and the other one is Nextcloud. And I will go a little bit more in detail on how we used it and how we converted it or managed it to our project. So first, the DataHub. So it's actually a platform for wet lab researchers where you can do a management of your data or your metadata, and it's being developed by Verdom and Elixir Belgium. And it's actually based on two elements. One is Verdom Seek, which is the flexible open source code, while the structure is being determined by the flexible standard model of ISO tools. This was made for multi-omics studies, but of course, as you could already see, we do much more than analysis, chemical analysis or metabolite analysis. We want to extend it now to also data sets of DNA assembly, creating strains, analysis of these, the performance of these strains at small scale and large scale. So if you look at the DataHub and how we wanted to build it, we started again from the end where the data will end up or is needed. And so one will be, of course, when we publish and we deposit the data in a database. And the other one is where we analyze the data, and especially then in function of these AI digital tools that we are making. And so we looked at what are the requirements of these databases of the AI tools. And there we defined these requirements to put them back into or to define how our DataHub should look like and how we should collect our data over there in which structures, making this possible that based on this data collection, we can do easily data analysis and then data deposition. So we started from here, looked at the requirements to build this so that we actually then can easily do these two elements. Based on that, you can see here, typically for here and to communicate between those elements, we can use API or either JSON or tab format. While for this one was a little bit more challenging, we really had to come up with our own experimental sample templates and so on, but I will go a little bit further in detail there. Also now identify the different studies or assay streams that we have within our project or within a biotechnology project. So how does this look like? The data hub, when we have access, has that program is now being redefined for us for deCYPher. And within this, when I enter and you see screenshots here, you see my data hub and I have different projects of this. And so for the projects here, there are 3 types of projects that you can add. This is composable elements, experimental templates, and then the work packages. And it's important to realize that the first 2 actually are feeds for the work package projects. And so the composable elements, that's what we decided on, what are the minimal requirements that we want to have when we do an experiment, and we have a strain there with a construct. What are the elements that needed to be given as a metadata? And so, for example, in here, what you will see in this projects of this composable elements, we will have the different parts, I would say, or types. And this could be genetic part, it could be the plasmid, it could be the strain, or the protein. And then the samples are actually feed-ins of these sample types. So it could be a strain entry or a plasmid entry. For each of them, and so at the moment we have 4, we defined with the group what is the minimal information required. And so when you want to enter one, you will have to give this element. So you can see you need a unique identifier, the name, the description, and so on, and when it's being entered for the strain, for example. Once you have these elements, then you also, besides the elements or the samples that you will generate, you also have the experiment templates that we are creating. And so we are creating specific ones for deCYPher. So they are being created also along the project. And so it could be, for example, a study of how the growth is, of how different sources can be used to optimize the growth. But also, for example, for us, in our case, these CYP enzymes, they have transmembrane domains. So we want to see if they really are being attached to the membrane. So we will also have microscopy data. So for each type of experiment that we will have, we define now what will be the experimental setup and which type of elements should be there. Once you have all these elements, then actually you can start doing the experiments and collect your data. And so we chose to store the data per work package that we have in our project. So for example, we have a work package on bioprospecting and functional expression of these CYPs. And then it's per partner. And then per partner, they will then add what is being called investigations. But these investigations actually are the same as the tasks being defined in the work package, in the work packages. And so for each work package task, we have an investigation. And then under this investigation, you get studies of the different experiments that you did with then the streams and the outputs or the relevant experimental outputs that will come. This looks a little bit like this. So for example, in our work package 2, we have a task 2.2, which is about localization of these CYP, about these transmembrane domains. And so you can see for Ghent University, we have different experiments running under it. And so, for example, you can already see here a hidden item. So this is something that is not visible for me. So for each level, that you can see, you can actually determine who can see it and who not. So you don't have to share everything with all the partners, because sometimes because of IP reasons or and other elements, you don't want to share all the data with all the different partners. That's more or less how the data management or the data hub is being created for us. And it's still, this was the sandbox, and this is now running really nicely. And so we are now transferring to the really product of this. Besides that, we also use Nextcloud, and that's just a tool that we use for the moment for data storage. And also if we want to share data without all the metadata, so like raw data. And for that reason, we decided within the project that we just divided it in, so each partner got its own folder, and each partner decide for his own folder and subfolders who will have view, read, write, or create permission or not. So these are the two main elements that we are using, and especially then the data hub reinforcing for biotechnology. So where do I see then links between what is going on in Bioindustry 4.0 and deCYPher as such? Well, something that I didn't discuss today, but we create a lot of different SynBio tools and methods. So maybe there is we work also on different localization methods, but also different genetic circuits and biosensors. So if people are interested, we are always open to see if we can collaborate or if we can help and exchange technology and tools. I assume as Bioindustry 4.0 and the digital twins that you are making and so on, the way of good practices that we have or that you have, that could be maybe also interesting to know, especially related then to the AI pipeline. But then, of course, the data management, the use of our Elixir resources, but of how we build it up, and the exchange of good practices could be something. And then, of course, and that's already happening, in our outreach, we have different workshops, but we have an artist in residence program, and actually it will be announced tomorrow. We are launching together an artist in residence program. And then in the end, this will also result in then a common art exhibition. And with this, I just want to acknowledge the consortium that we have and all the people involved making this, because they are the experts, especially what I presented today, and then of course the EU of funding our program. And I think with that, I come by the end of my presentation, showing you a little bit more about the data management and AI tools that we use.